It's just a soft gland. It's behind the stomach. It has both endocrine and exocrine function. When we start the course, we do endocrine, and I talk about the islets of Langerhans. Remember that? And well, that's the endocrine function. The exocrine function uses that main pancreatic duct. In the study guide, I always ask where it is. It's located in the space called the lesser sac behind the stomach. Note the pancreatic duct because that's where it'll dump its juices into the duodenum. And let's kind of look at it here. There's a picture of it associated with the duodenum. The main pancreatic duct is the white structure and empties into the duodenum. And the physiology that we're going to talk about are mostly the exocrine pancreas, because I do want you to study that pancreatic juice, which contains bicarbonate ions and pancreatic enzymes. So the pancreatic duct is a duct for pancreatic juices. And those juices are comprised of a bicarb-rich juice, bicarbonate-rich juice, as well as an enzyme-rich juice. The bicarb-rich juice, well, that will help neutralize acid. The enzyme-rich juice will help digest, well, everything. Fats, proteins, carbs, nucleic acids. Fat, pro for protein. CHO for carbohydrates, nucleic acids. There's enzymes for all of that digestion. And this juice, you produce about 1 to 1.5 liters a day. It's mostly water, but again, mixed in there is bicarbonate or a mixture of enzymes. Now, I have some of the enzymes listed there. Proteases, amylases, lipases, nucleases, that digest all those things I've listed. And as an example of how they work, I'll look at the protease activity. So what happens is these juices, these enzymes, are released into the lumen of the duodenum in an inactive form to prevent autodigestion. And when they reach the lumen, they'll become activated by this membrane-bound enteropeptidase. So that's a very important concept in digestion. You release things in an inactive form, so that way you don't digest along the way as you're being secreted. All right, so let me erase this. All right. Release pancreatic enzymes in an inactive form. Prevents autodigestion. So in the example for the proteases, trypsinogen. That's the inactive form. It does no harm as it's secreted. But when it reaches the lumen, there's a membrane-bound enteropeptidase that will activate it to its active form. It'll convert it to its active form, trypsin. Membrane-bound. Well, it's important to note that it's membrane-bound, so it doesn't seep into any structures where it can be activated prematurely. Membrane-bound. Enteropeptidase. Okay, that will do the conversion of trypsinogen into trypsin. That's the active form. And what you can see from the picture is then that trypsin in turn will activate other proteases. So then digestion can begin in the lumen of the digestive tract and not anywhere else. So I'll just say activates other proteases. And so this is what we understand of how all the pancreatic enzymes work. When they're secreted, let's look at the main structure of the glandular tissue in the pancreas. Okay, so remember, this is exocrine function here. So this is a good shape to look at right here. Pancreatic activity. So title this pancreas, exocrine function. You've got those bluish duct cells. Then you have those orangish acinar cells. This acinar structure describes the shape. It means mushroom shape. Well, anyways, these orange ones are the acinar cells. And the blue ones are the duct cells, and they secrete different things. They're targeted by different things. Secretin, as we said before the break, it's released in response to acid. And, well, it's going to target the pancreas, but specifically it targets the duct cells to secrete the bicarb-rich juice. So when we get acid in the digestive tract, that's a trigger for the release of secretin. One of the things it does, it targets the pancreas. It'll target these cells specifically to get your bicarb-rich juice. Okay. Everything with CCK. CCK, in response to fat, it will be released and it'll target these acinar cells and they'll secrete the enzyme-rich pancreatic juice. So, in response to some fat in the food chain, trigger for the release from the intestine, CCK, targets the acinar cells, and that'll stimulate the release of those pancreatic enzymes. And it's a combination of the bicarb-rich pancreatic enzyme-rich juice that are the pancreatic juices secreted daily, about 1 liter to 1.5 liters. So that's the exocrine function. So basically, the CCK will balance the fat digestion in the secretin. When it's released, you'll get the bicarb-rich juice. That'll help you with the acid neutralization. But let's, I put another slide in here. So we get the secretin and CCK for the bicarb-rich pancreatic enzyme-rich juice. But let's not forget the endocrine function. What I had mentioned earlier was those juices use the main pancreatic duct. However, the endocrine function is stimulated by GIP. GIP is released in response to glucose, and it'll stimulate the release of insulin for endocrine function. So we talked about insulin before, but it's important for digestion, so I want you to still know that. So let me draw some cells outside of the acinar structure. So that's a little pancreatic islet. We got the alpha cells, the beta cells. The beta cells secrete the insulin. So in response to glucose, those levels rise. And what happens is, from the intestine, you'll get GIP. GIP is one of the things that can help stimulate the release of insulin to help lower blood sugar. All right, so, but that's endocrine function, not exocrine function, just to be clear. Okay. The liver is next. All right, here's some notes on the liver. It's the largest solid organ in the body. They say it's about 2% of your body weight. It's a soft, reddish, brown color. In our cadaver, it's kind of rock hard over being years of just kind of being in a cadaver. But it receives a lot of venous blood that returns from the GI tract. So we call that portal blood because one of the main veins that receives blood is the portal vein. The portal vein came from basically your guts, your intestines. And gas exchange has already occurred. So it's nutrient laden, but oxygen poor blood. So it needs a second blood vessel to give it oxygen-rich blood, the proper hepatic artery. It's a storehouse for glycogen, but its main job, as I said today, for digestion, it helps to secrete bile. An important agent for digestion. It can be stored and concentrated in the gallbladder, between meals when you're not eating. On the anatomy, there's some lobes here. So here's a frontal view of it. You have some peritoneum over there, covering it. Kind of dives in like that, covering it here. Okay. Well, anyways, let's just talk about the lobes first. So I have 4 lobes listed there: right, left, caudate, quadrate. Now from this view, you can only see right and left lobes. So, which is the right side as you look at the man? That's the right side. So the right lobe is the big lobe. there's the liver isolated from the body. So that's kind of like the front view, and well, you got two ligaments here. You got ligament of teres, round ligament, and falciform ligament. Remember, this is our bare area, our bald spot, because it's not covered with the peritoneum. Well, anyways, the falciform ligament is this part right there. Falciform lig. The round ligament, or ligamentum teres, same thing. Teres means round. So maybe you just want to call it the round ligament. It seems like it'll be easier to remember. But when you see ligamentum teres on the key, don't ask me what it is. All right? Teres means round. Same thing. Now, I don't know if you remember this. A little blood vessel in there. Contains obliterated umbilical vein. Okay? It's in there. So that's the round ligament on the bottom there. The falciform ligament, what it does is, the falciform is attaching to the anterior abdominal wall, on the inside of your body cavity there. Falciform leg attaches to the inside of your anterior abdominal wall. I'll just say anterior ab wall. So that's the next slide there. So I'm pointing to the round ligament in two locations. On the liver, which you gotta know, there's falciform right there. But this is also falciform right there, attached to the front body wall. At the bottom, that's the round ligament, and you can see how the umbilical vein goes down there. Do this. So at the very bottom is the round ligament, and it contains the umbilical vein right there. Umbilical vein is running down here. Umbilical vein. And you have two umbilical arteries coming up to it. One umbilical vein, two umbilical arteries. Remember I kept saying that? Over and over again. Here's one umbilical artery, two umbilical arteries, two umbilical arteries that go up, and they go through what? They go out the belly button. Okay, so that's the inside of your belly button. All right, this is the inside of your anterior abdominal wall. Okay, usually you don't look at that. You usually look at the outside. But anyways, that's where that's coming from. So no falciform, no round ligament. The round ligament contains the now useless umbilical vein. But it was well used when you were in utero, inside mommy. Okay. Well, anyways, I think that's cool. I can show this to you on cadavers too. The inside of your belly button. You're just staring at the outside of it your whole life. Okay. This is the underneath view of the liver. I don't know what it is. So it's like we got more or less that kind of shape. You got the inferior vena cava there. It's all part of it. You got like an hepatic vein going in there. IVC, inferior vena cava. Hepatic vein. This kind of doorway here. So on the underneath view, there's our ligament teres, our round ligament on the bottom. Round ligament. And just to reorient you, this is the left lobe, that's the right lobe from the bottom. But we can see our other two lobes. We can see there's a little tail lobe right here called caudate, a four-sided one called quadrate lobe. Draw them in. Caudate lobe. Quadrate lobe, four-sided one. And right on the right side, right next to the quadrate lobe, is the gallbladder. Now the gallbladder is just filled with concentrated bile, but you can see it has a little duct. It's going to exit right there. So this green pouch, gallbladder. Now this little opening here actually has a name. It's a doorway. It's a doorway to the liver. It's called the porta hepatis. Porta hepatis. I'm trying to see if that term is up there somewhere. Porta hepatis. No, I don't see it. Well, anyways, let's label it on our picture. Call this opening right here. That whole area where things go in and out. Porta hepatis. Means doorway to the liver. Well, bile is going to come out right there. That's the hepatic duct. Okay, the bile duct, I should say. Bile duct right there. Those are these two other blood vessels. One's a portal vein. The other one's proper hepatic artery. Portal vein right there, and then proper hepatic artery. That one there in red. I'll just put it over here. Proper hepatic artery. And then I'll draw a blue structure. It's nutrient-rich blood, but oxygen poor. Okay, so that blue one is going to be portal vein. And then that green structure, bile duct. Those are the three contents of the porta hepatis that you should know. That's why I kind of drew them next to porta hepatis. That's the anatomy I want you to know. Things go in and out of the liver. So the liver's job is to produce bile. So bile's coming out, right? Bile coming out. But blood going in, this blood from the portal vein, that blood came from your guts. It's got all the fugate. And it goes to the liver first so you can process it. It's very important to understand. Anything you stick in your mouth, it's going to your liver first. If it's McDonald's, if it's vitamins, if it's supplements, if it's vegetables, whatever you stick in your mouth, it goes to the liver first, okay? Because the portal vein. Then, well, if blood goes in here, blood also goes in there. That supplies the oxygen-rich blood. So it's kind of like, remember the umbilical vein for the baby? There was just one of them. And that one umbilical vein had oxygen-rich blood and nutrient-laden blood for the baby. In the adult, it takes 2 blood vessels to accomplish what one umbilical vein did. So we have 2 vessels, one for nutrient-laden blood that the liver can process, and one for oxygen-rich blood from the proper hepatic artery. So it's very important. Blood going in, blood going in. Go to where does blood come out? Hepatic vein. Once blood comes out there, the liver's done processing it, and it goes to IVC for regular systemic circulation. Okay? So blood going in, blood going out. So in terms of naming, portal vein is sometimes called hepatic portal vein, but sometimes students confuse that with hepatic vein, and you think you deserve partial credit because you were close. But functionally, these vessels are completely different. One, blood's going in, one, blood's going out. So no partial credit. Because the function is so different. All right, well, anyways, moving on. Okay, so this slide, I'm just reminding you the contents of porta hepatis. We listed them on the board there. Portal vein, proper hepatic artery, bile duct. So those are our pictures side by side. We got models there. Can you hand me that, Andrew? A model of the things. So on this model, they got it on the stand. Looks like how I drew it here is like that, right? Yeah, so in the body, this one is like, actually it's this way. So the stand faces you, just so you can orient yourself. Okay, so that's a good model to look at. What else we got here? That's a close-up of the porta hepatis, gallbladder, bile duct, proper hepatic artery, portal vein. So the gallbladder, the biliary system, as we call it, is a bladder and a bunch of ducts. It's not much to look at. It's a pear-shaped sac. I mostly notice it because it discolors the entire region on a cadaver, that greenish color. It's a sac that lies along the right edge of the quadrate lobe. It's bluish green, can hold about 30 to 60 ml. Well, there's a picture there. You remove left part of right lobe, and you can kind of see it there next to the duodenum there. So that's the anatomy. Let's map out all these different ducts, because you do have to know that. Let me erase this here. Now the liver, it produces about 900 ml of bile a day. However, not all of that is reaching the duodenum. You know, some of that is produced in between meals. So maybe in between meals, it's about 450 of that 900 a day. Say between meals and quotation marks. It varies, but the point is that, well, if you're not eating, you have to divert it to somewhere. And usually because when you're not eating, that sphincter is shut. The resistance is such that it'll wash backwards and be stored in this gallbladder. Okay. So the liver production, this bile produced by the liver is considered dilute. But when it's transferred to the gallbladder, the wrinkled surface, it helps to store and concentrate it. And so bile secreted from the gallbladder is called concentrated bile, not dilute bile. So when you stimulate the gallbladder to be contracted, the bile that reaches the duodenum is actually a mixture of dilute and concentrated. So about roughly 500 ml bile reach the duodenum every day. And that 500 ml is a mixture of dilute concentrated. A mix of dilute concentrated. All right, so in terms of the anatomy, let's kind of map out the different ducts. So at the top, there's a V shape here. You have right and left hepatic ducts. You can assume they're draining bile from right, left lobes, respectively. So that one, right hepatic duct, left hepatic duct. They merge to form common hepatic duct. Now the common hepatic duct is merging with the cystic duct, which comes straight from the gallbladder itself. Form bile duct. And I already drew it earlier, but the bile duct merges with the pancreatic duct and dumps into the major duodenal papilla right there. Okay, but those are some of the ducts I hadn't mentioned before. So the thing about the gallbladder is it doesn't have a sphincter, but it has this spiral-shaped valve which well, it just helps keeps the bile in there without having to have another sphincter. That has a sphincter, that right there. That is just a spiral valve, so stuff gets in, it's hard to get out, and it concentrates there. And so remember, when you squeeze this thing, you have to relax there so that the resistance will be decreased and allow the bile to move out of there. All right, so you can't live without a gallbladder. I mean, gallstones kind of form there and clog this thing up. And it has to be removed in emergency surgery, but you can live without it because you have plenty of bile being produced by the liver, but it's the dilute kind, so it makes it more difficult to digest fats. In terms of the anatomy, those are pictures you can study. In any liver model you get, if you can see the gallbladder, you can orient yourself because the gallbladder is always associated with the right side. So if that's the gallbladder and this is the right side, then you know that's the right hepatic duct, therefore that's left hepatic duct, common hepatic duct, cystic duct, and right at the tip would be bile duct. The purple is portal vein, and the red is proper hepatic artery. So again, no matter how I turn the picture, if you see the gallbladder, you're oriented. That's the right side. So that's right hepatic duct, left hepatic duct, common hepatic duct, cystic duct, bile duct. There's portal vein, that's blue. That's a proper hepatic artery. So I can't see it there, can't see it there. Oh, here you can see it, a little blurry. Well, anyways, okay, there's the gallbladder. So what side is that? The right side. So that's right hepatic duct, left hepatic duct, common hepatic duct, cystic duct, bile duct. The purple is portal vein. What's the red? The artery, the proper hepatic artery. On this figure, there's the inferior vena cava, and there's, excuse me, hepatic vein. We'll talk about the liver log, and we'll pick it up here next time. The rest of the time, just go ahead and study the models, and you've got some more things to study now, and I do want to remind you of your quiz on Monday. Will be the first